Welcome. In the next hour, we're going to learn about the next steps to conservation in regard to citizen scientists. It's the next step for wildtrack, and it's going to be a very cool endeavor, I believe, and I'm very excited to be here to introduce our scientists. I could go through their CVs, but that would take a very long time, so I'm not going to do that. You can Google these guys and learn a lot about them if you'd like, and if you've got a lot of time. But for now, I'm going to be brief, and I'm going to introduce Dr. Stuart Pym, who is the Doris Duke Professor of Conservation Ecology at Duke, and the wildtrack founders, Sky Alibi and Zoe Jewell. They are going to talk to you about what they're working on, and I believe it's very, very cool. My name is Diana Levy. I work for the Jump Division of SAS, and I am on the board of directors for wildtrack, and I'm a very big fan of what they're working on. So with no further ado, I'm going to ask Dr. Pym, please come join us and talk about what you're working on.
Thank you so much for coming and listening to us today. I'm Stuart Pym. I'm a professor at Duke, and I also have an absolutely wonderful title of extraordinary professor from a university in South Africa. I think it means it's extraordinary that they don't pay me, but nonetheless, it's a privilege to work in such a rich and interesting country. When Al Gore, in An Inconvenient Truth, said a mass extinction crisis with rates of extinction now 1,000 times faster than the background rate, you might have wondered from whom he got that, and the answer is from me. It would have been nice if you'd have said, you know, my buddy Stuart Pym, but you know, in academia you take publicity any way you can get it. So I'm particularly concerned with how fast we are losing biodiversity. It's running 1,000 times faster than it should because of the various things we do to the world, to the land, to the ocean. And we know roughly what the main problems are. The rate of extinction is increasing. Of course, extinction is irreversible. It's geographically concentrated. Where species are makes a lot of difference. And tropical deforestation turns out to be a major driver. What I want to tell you this morning is that what we need to do about that has a lot to do with understanding where species are, so we can set our priorities, and who the individuals of those species are, so we can manage them. So the first point I want to get across is this is a problem that requires staggering amounts of data, which makes me feel as if I've come to the right place. Part of the problem is we need to know what we're doing to the environment. We need to understand land use changes. And the technology of satellite imagery has made that ever more possible and ever more easy to understand. In the New World, in the Americas, the concentrations of species are in what we call tropical rainforests, tropical moist forests. Brazil has most of the species. The Amazon is a huge area. It used to look like the area at the left, a huge block of continuous forest. But if you look at what it is now and you color code it by how far the forest is away from an edge, you find that it's being eaten away. Even worse is the coastal rainforest of Brazil, an area the size of California, Oregon, and Washington, where almost all of the forest is gone and what remains is in pieces. Now, with satellite imagery, we now have an ever better and continually improving understanding of what we're doing to the planet from space. But that's not the only problem. We need to understand where something like 10 million species are. distributed across 130 million square kilometers of ice-free land surface, and we need to understand how their ranges, their geographical ranges, are changing over time. We have a need for prodigious amounts of information on species that you cannot identify from a satellite image.
The number of species of birds and mammals and amphibians we can now map out with some considerable detail. These are birds, and they show that the greatest numbers of species are in the tropical moist forests of the world, a pattern that covers all the continents. The interesting and unusual aspect, however, is when we begin to look at species in terms of their geographical range sizes and look at the species that have smaller than the median range. Those distributions are profoundly different. They are concentrated in special places, what my colleague Norma Myers calls hotspots. And the northern Andes is a hotspot with extraordinary numbers of small range species.
Now, the broad patterns, the fact that places like the northern Andes and coastal Brazil and the Philippines are places that have a large number of species with small ranges and are geographically concentrated and at risk of extinction. We understand that in some general detail. But you know what? We ought to be managing our natural heritage on more than just birds. You know, we need the bees and the butterflies and frogs and flowers and everything else. And we have very, very poor information on that, except that has changed dramatically because of a device that could have been designed to be the best survey instrument ever for biodiversity. We can encourage people to go out in the field and snap a picture of a frog or a butterfly or a flower. And even if they don't know what it is, they can upload it to a website, iNaturalist, which was born in my lab at Duke. I'm very proud of that. And people will identify it. People are passionate. There are groups of people out there who love orchids, who love dragonflies, who love frogs. And that figure on the right is one of the most compelling exponential growth figures I've ever seen. It took us about 4 years, 5 years for iNaturalist to get its first 1 million observations. The prediction is by 2020, we'll be getting 1 million observations of species per week. We are getting to put together a compelling story of where species are, how their ranges are changing as the planet warming, and it's all driven by amateurs. It's all driven by people who snap a picture of something, upload the information onto iNaturalist. And that's just a page from a couple of days ago showing, you know, the hundreds of people who had uploaded an observation to iNaturalist within the last few minutes. There's something else we need to do. We want to know who the individuals are of some of these rare species. We don't just want to know where the species are, we want to know who it is. Is it Zoe? Is it Sky? Is it, you know, any one of you? We quite literally want to know the lion by its paw. I know that's not a good translation of the Latin, but you probably recognize the famous quote. We want to be able to identify individual lions. We want to be able to identify individual pandas. Pandas are miserably difficult to count. They're secretive, they live in remote places. People are identifying pandas by their droppings, by what my Chinese student says very engagingly, panda poops. You know, if the poop's that big, it's a different panda from if it's that big. And that's not a very good way of identifying pandas. We can't even sex them in the field. We assume that they can. How do you tell the difference? Well, that's where ConservationFIT comes from. And I think that this effort, which is based here at JUMP, is the most exciting technological advance, because we can now begin to do conservation involving lots of people, lots of amateurs, combining really basic elementary natural history with the kind of very, very sophisticated statistical analysis for which you all are so famous. And without further ado, I'll hand it on to my colleagues.
Thank you very much, Stuart. I'm afraid I've disappointed Stuart once again by not being properly attired. Sorry, Stuart. What I'm going to do is to basically just give you a very, very, very brief run through what it is that we're actually doing as far as conservation work is concerned in JUMP. And then Zoe will come on next and talk about the way in which now we're incorporating SAS into the whole thing. So basically, ConservationFIT and wildtrack will be encompassing both JUMP and SAS. Now, I'm just going to use a very, very simple example to demonstrate the sort of problems that we're facing in the world today. These are 3 species straddled across 3 continents. And they're highly iconic, endangered. But when you begin to ask a simple question, how many are there and where are they, our information on that is woefully inadequate. Let me just demonstrate that using one example. The cheetah is perhaps one of the most well-studied, one of the most well-researched big cats in the world today. Now, all I want to do at this stage is to just point at the map of Africa and ask you to hone in on those 3 arrows. Now, I'm colorblind, so I do sympathize with people who are colorblind, but if you can make out the red, that is actually what the present-day distribution is. And in some places, you can barely make out the red spots. And then if you hone in on the yellows and the purple, huge swathes of areas of Africa where we just do not have any idea what's going on as far as the cheetah population is concerned. So it, in a way, dramatizes the whole impact of our lack of knowledge about animal distribution and numbers. Why do we need this data? Now, one can come up with all sorts of reasons. I've actually summarized this in 3 small sections. One is, in order to be able to get data, in order to be able to drive scientific conservation strategies. Okay, if you don't have that data, you don't know if your strategy is working or not. Secondly, there's a huge trade now in endangered species, illicit trade in endangered species. And that's funding terrorism, that's causing destabilization in many, many parts of the world. We need data from those areas where this is going on. And finally, human-animal conflict. You and I look at elephants and we gope at them and so on. But there are parts in Africa where elephants are a nuisance. We need data on that to be able to mitigate that conflict that's going on. And that's why there's a need for data. And with that at the back of our minds, years ago, Zoe and I set out, gave up our normal jobs, as one does, and ventured forth to form wildtrack. And the whole idea was to come up with monitoring strategies which would provide that sort of data. And we are concentrating our efforts on non-invasive techniques, and there's a huge story behind that, which I won't bother to go into now. But the idea is to try to disturb the animals as less as possible so that the data that we're getting from these non-invasive techniques are reliable data. We partner with many, many organizations. We are based here, in case you don't know, we're based here at JUMP. and we are also principal researchers. We are associate adjunct professors at Duke, and also have a connection with Australia. We've got all these different connections. Some of them are government organizations, some of them are funding organizations, many of them research organizations. So we partner with many, many different organizations. And the idea is to have this interaction so that we begin to understand what the problems are in those different areas and how we can actually deal with them. Why footprints? Well, two simple reasons. One is that footprints and tracking formed the basis, perhaps, of our evolutionary history. If it hadn't been for tracking and footprints, perhaps we wouldn't be where we are today. That was actually fundamental to the whole evolutionary process of humans. And secondly, footprints are everywhere. They're ubiquitous. You find them everywhere. And I bet you from now onwards, when you go out into the wild somewhere, you will not be looking out for that bear, which might be at a distance, looking on the ground where there's a hell of a lot of evidence, which we are not gathering. And of course, it's highly species specific. Footprints come in different shapes and sizes. And we have now got databases on a large number of species. We have projects in many parts of the world. We now have on our books something like 25 different projects. Some of them are projects that we've dealt with. Some of them are very, very active projects, particularly in China and in Africa, and now in South America with the jaguar as well. And I'm just going to briefly talk to you about the way in which we do the analytics in JUMP, and then Zoe will come and take over and talk about how we're dealing with deep learning in SAS. We have a couple of, well, three actually projects in China with the Amur tiger, the Amur leopard, and the giant panda. There's me pretending to be a giant panda, not very successfully. They have these huge enclosures, and if you want to go into one of these enclosures, you have to put on one of these panda outfits. It is never cleaned because the scent will disturb the panda, so you can imagine that the conditions were somewhat ripe, to say the least. And there's Zoe when we went to Northeast China, where we're studying the Amur tigers. And I'm just going to do a very, very brief run through the analytics that we do in JUMP. I'm not going to do a live demo of JUMP for two reasons. One, it takes a bit of time, and the other reason is Zoe claims that I usually put everyone to sleep. Okay. So we've got all these different species now. I mean, JUMP, I'm sure you'll recognize the home window for JUMP. We've got all these different species that we have databases for, and I'm just going to run through the way in which we do the analytics. This is our home window for the Amur tiger. It's all species specific. For every single species, we have a different FIT setup. We can do image feature extraction. So I just pull on the window, drag and drop images in there, do all the enhancement, all the manipulation. We extract the metrics, and then we do the analysis. We run whole back trials to test the model. And I just, very, very quickly, if I can mention, the model that we've based the whole of FIT on is the notion that there you are out there in the field, you come across a trail, and then you walk a couple of kilometers away and you come across another trail. And the big and central question is, are those two trails from one individual or two individuals? And that's exactly what the whole analysis, and that's why we call it a pairwise data analysis. We do validation trials, and then we've incorporated that into a classification dendrogram, which gives us an estimate of the numbers. And then using that, once we have actually optimized the model, we then use it for unknown individuals. These were trails that were sent to us by our Chinese colleagues, and they said, well, we picked up all these trails from this area. How many pandas are there? They sent us 13 trails. This is just one small trial that we ran. Do we have one panda or 13 pandas, basically, or whatever, any number in between? And we ran the whole system, and we came up with seven tigers, sorry, bigger one, tigers I'm talking about, 7 tigers, and we actually determined the sex as well. We can actually discriminate sex quite easily in the Amur tiger. we can actually discriminate between Amur tiger and Bengal tiger, just as an interesting exercise. And as I said, we can actually discriminate sex in the Amur tiger and in the giant panda as well, very, very successfully. And finally, we've also got mapping in JUMP. So it's really a one-stop tool that takes us from actually dragging an image into JUMP to actually producing a location point on a map. So basically, FIT, footprint identification technique, is non-invasive. We don't resort to these types of techniques. It's cost-effective. It's pretty accurate. And as I said, it's a one-stop monitoring and mapping tool. And I'll leave you this final slide and ask Zoe to take over.
So I guess the place we go from here is we've got a technique that works really well for identifying at species, individual, sex, age class levels. But we're hugely lacking in the data that we need to be able to figure out where animals are on that map. So there are a number of challenges, and with those challenges, there are opportunities that come for all of us in being able to participate and drive this project forward. So I'd like to talk a little bit about that. So the first challenge is collecting the data. Now, up until now, we've worked pretty much on a one-to-one relationship with all those projects you saw across the world on the map. We help them gather data, they feed us the data, we give them individual feedback, they come back to us with images, we say that looks okay, but try a little bit this way. And so this has been a very slow and kind of customized approach for each project because we've regarded it partly as a developmental phase, and that's been great. But we're now in a position that we have the opportunity, as Stuart has said, with cell phones and with other pieces of emerging technology, that we can really begin to collect data on a massive scale. And with that comes some challenges. So this slide really is designed to show the new areas that we can hope to collect data from. I don't know if this guy is working. Let's see. Anyone see a pointer there? Oh, there it is. There it is. So drones. Drones are now becoming hugely popular. Lots of people have drones. Lots of people fly drones in parts of the world you wouldn't necessarily think that they do. And using drones, you can see trails. And if you zoom in close enough, you can begin to see species and individuals. So drones are a potentially very valuable source of data for us. The market is growing incredibly fast. Our 16-year-old has 2 drones and is busy flying them and adapting them and doing what we ask him to do with them. So we've hijacked him, and we're hoping to encourage a whole lot more. Recreational visitors, 8 billion visits were made to recreational areas in 2014. That's a staggering number of visits. Now, if you could actually engage, 0.5% of the people who go to these protected areas to collect data with smartphones or even drones where they're allowed to, we'd have masses and masses of data coming in from there. Indigenous experts, we developed our footprint identification technique with indigenous trackers. They're the people who said to us, why are you waving this radio transmitter around in the air when you could just be looking down, all the evidence that you need is in front of you? And they would laugh at us. So we thought, well, there must be something in this, and that's how we developed the footprint identification technique. But these guys are out there. They're seeing things all the time. They've got the most fantastic powers of observation. So if you give them smartphones, and you say, OK, when you see a footprint, please pick it up, another huge source of data. Stuart talked a little bit about satellite imagery. That's again, the global market in satellite imagery is expanding very rapidly. The resolution of images is improving all the time. We can begin to map out habitats and ground truth what we see with individual observations. We're also engaging with the captive animal community through zoos. For example, the American Zoological Association, zoos and aquariums, their species groups, their taxon advisory groups. And these captive animals actually allow us to develop the reference data sets that we need to develop the algorithms for FIT. And I think what they find particularly interesting about this project is not only that they can provide data for us, but that in a sense, their captive animals are helping conserve their free-ranging counterparts. So it's a very nice connection point. And there are 175 million visits per year from people to zoos just in this country. So again, that's a huge number of people that we can engage, that we can then ask to help collect data. And sitting right in the middle of all this is, where's the pointer gone? Sat and jump. There it is. Sat and jump, basically, as a conduit for all this data coming in. How do we manage it? We'll talk about that in a minute. So I want to just show you a little bit about how we're going to manage it. And that is our new website, ConservationFIT. Please bear with me as I do this. I'm a left-handed Mac user, and this is a right-handed PC, so anything could happen. But we are going to try, see what happens. OK, there's our homepage. So please, when you go home, take this for a test drive, have a look at it. We developed this specifically to be able to engage all these groups of people around the world to give us data, to feed us data. So on this site, if I can figure out how to use this, there we go. On this site, on every single page, and there's lots of other information on here, I won't go through it all, but on every single page there are a series of interactive buttons on the right-hand side for people who are interested in helping with the project to engage with. So there's social media, there's a forum, there's a blog, there's contact us. There's a button to show you how to use the site. So it takes you through how you take images, which parts of the site do what. There's a site that shows the observations we've got. There's some instruction on how to take images for FIT. We have a little protocol. It's not really rocket science, but it takes a little bit of practice. So you lay a ruler down on the ground when you see the footprint, and you take the footprint from directly overhead, and you have to be a little bit careful about where you're standing and not tread on the footprint and stuff like that. So all the instructions are in there to help people be able to do this. And then the one thing I'd like to show you, and Stuart already showed a little bit about this with iNaturalist, is that you can upload images using iNaturalist. So when you click on that button, up comes our special project in iNaturalist. And if you click on this great big red banner, Add Observations, up comes a little form which you can fill in to say where you found your observation. So let's say you're out in the backwoods and you see a carnivore print, you think it's not the neighbor's dog, it's a wolf come to get you. And we have these emails sent to us all the time. I found big dog prints, or no, big carnivore prints around my car. What is it? And it's usually the neighbor's dog. But if you think it might be something that's not the neighbor's dog, you snap it up, you upload it here, and it tells that this gives us all the information we need about that image. You can choose the files from your... See, I've got no control over this thing. You can choose the files from your desktop. You can upload them. It'll put a point on the map. You can describe what you've seen. We've got some special fields down here that we're asking people to fill in. And along with the image that you've uploaded also comes all the metadata. So the beauty of iNaturalist is that you can download all this data, and I'll show you that in just a second. So let's imagine you've uploaded your print, and in fact you haven't been next door, you've been down to Namibia and you've seen a cheetah. We'll click on this and up comes the information. That's actually my cheetah print, but you can imagine that it would have been yours. And you have 29 photos here that we've taken from this cheetah in Namibia. So all your images come up, and you can click on any one image to see a little bit more of it at different sizes. And not only can you do that, but you can go out a little bit on this site and you can see who else has picked up cheetah images in Namibia. So now let's say you found some, but you wonder if the images from that cheetah are the same from, say, somebody who's seen a cheetah up here. Let's click on one of those, see what they've seen. They've seen a cheetah. Now that person's made lots of observations. All their data is on there. You can find out a little bit about who they are, where they've been. We could contact them and say, let's see if your cheetahs match up with our cheetahs. So the potential for mapping is absolutely huge. And if you want to go one step further in that process, you can actually look to see, this is information about the top identifiers for cheetahs. And down here is a map. How do I get down here? Oops, no, it's not doing that way. Anyway, I think, there we are. You can see the map of Africa with just some of the observations that people have made about cheetahs. Now, I would say 99% of these observations are pictures of cheetah. And some of them are at a distance, some of them not too great, some of them are really good, but very, very few are footprints. Now, we calculated the other day that if a cheetah is walking from point A to point B across the desert, let's say it's going 100 kilometers, which is not unusual for a cheetah, in that period of time, that one cheetah in that 100 kilometers will make 400,000 footprints. So we're talking about 5 orders of magnitude more data here than we would get from one cheetah observation. Now, granted, cheetah, footprints may not be quite as easy to see as the whole cheetah, but still, even if you only see a fraction of them, that's a vast amount of data, and it's being renewed every day as the cheetah moves on. So if you imagine that map could be absolutely smothered in data points, and we could have them all connected up, and we can download all those data. So if I go back to, let's see, go back to this bit. We can actually download all this data using, I think we have to go back one more. Here we are. We can actually export, we can do queries on all the data, not only our own data, but everybody else's data. So we can bring all these images down with the metadata. We've got a vast repository of information to start mapping numbers and distribution. Even if all that somebody sees is a picture of an animal and you can identify the species, or one footprint, you can identify the species, that's still mapping species before we even get down to the individual levels. So there's a lot of information out there. If I just, what do I do? Just minimize this maybe and go back to this. Will that work? Brilliant. So after iNaturalist, and we assume that lots of people are going to be sending in this data now, we've launched this project, how do we manage these very large volumes of data? Now, this was something that put us into a minor panic when we started setting up this project, because we thought, well, this is great. What's going to happen when we have 400 footprint images coming in an hour? How are we going to manage those data? And luckily, we started thinking about the possibility of using deep learning. And there's a deep learning group here at SAS that we've been doing some preliminary investigations with on these data, just using some of the data types that we've already got. And there are very many different types of data that are amenable to this technique. But one thing I think that our data has that is unusual is a degree of complexity. When you think of a fingerprint, and everybody has a fingerprint, you can put your fingerprint on a piece of paper and you'll have a pretty clear image, black lines on a white paper. And even if you only get part of your fingerprint, it's still going to be the same fingerprint. Now, when an animal puts its foot down, every time it puts its foot down, you're getting a different print. And that's going to vary with very many different variables. So in terms of the difficulty for a computer in trying to understand these images, it's really multi-layered data. So let's just talk a little bit more about footprints. And here's a picture of a drone, which is one of the types that we're deploying for short-range reconnaissance at the moment. One of the things we'd like drones to be able to do is help us just find trails. So when we're out in Africa, we might be walking 20, 30 kilometers a day. It's blisteringly hot, it's hard work, it's dusty. Wouldn't it be much easier if we could sit in the back of the truck and have the drone fly a nice little circle around the truck and pick up data for us? So this is the first target of our drone flying. We're going to be in Namibia in 4 weeks' time. We're going to take the drones out and start using them to collect the data. Where's this pointer gone? Oh, there it is. Now, if you've got a drone, these are off-the-shelf drones with fairly limited camera power. If you've got a more powerful drone, a bigger drone that can carry an SLR, you can start identifying species on the ground. So you can home in on that trail. You can even find which species made the trail. And if you've got a really high-resolution camera, you can actually identify individual footprints. So you can fly your drone over, you can have it come down, hover over the footprint, pick up the footprint, and you've got your data. And all in a fraction of a time it would take a person to go out there and track those animals. And our colleagues in northeast China are particularly interested in this because they literally wade waist deep in snow looking for tiger prints in the bitter cold of the Chinese winters. They're super hardy field biologists. And so they're very interested in using drones. We have a challenge there in that the drone batteries don't like super cold. So we need to be able to work with people to try and develop slightly better insulation for those batteries so that they'll work. Now, apart from the fact that, footprints in themselves are challenging, the substrate will vary a lot. And every time the substrate changes, the image of the footprint, the look of the footprint will change. So for a deep learning group, this is another thing that has to be taken on board. How do you deal with all these different substrates, ranging from snow to mud to sand to gravel? These are all tiger footprints, and they just vary hugely in different substrates. And then we've got the challenge of different species. So we've got two sets here, set on the left from jaguar and set on the right from puma. To an average person, they look pretty much the same. And to us, they look different because we're used to seeing them. But again, for a computer to be able to figure out what's puma, what's jaguar, that's a great challenge. And we're working, again, with the SAS group on this. And as I said earlier, there's a big difference within each trail for footprints and between animals. So one of the nice things that our FIT technique does, it actually encompasses that difference within the range of one individual and compares it with another individual. Deep learning will have to learn how to do that. And then we have stripes. We have stripes and we have spots and we have all kinds of patterns in the animal world. This is a range of tiger images that, again, came from our colleague in Northeast China. There have been several programs that have been set up to identify tigers from their stripes with varying degrees of success. Usually a certain amount of manual marking is required. But again, we're hoping that with computer learning, we can actually improve on that. Here's a nice local example. North Carolina, box turtles, which range all along the eastern seaboard of the United States, and they have these individual shell patterns. So the Piedmont Wildlife Center asked us if we could help them identify individual turtles for a citizen science project, that people could go out, snap a turtle, and put the images up here, and again have a deep learning approach. It's working in footprint identification technique, but we haven't yet checked it out with a deep learning technique. And lastly, another lot of images. This is an interesting project. This is trying to figure out what Amur tigers eat and what Amur leopards eat. Now, we all know that what goes in through the front of the tiger comes out through the back of the tiger, as nicely demonstrated up here. And what the Chinese scientists are doing is taking hair from the prey species that's been eaten by the tiger, and actually identifying the species that the tiger or the leopard have eaten from the hair structure. And this is the medullary part of the hair from these different prey species. And this is really kind of this is a tough challenge, I think. They look superficially very similar. But if you could process them rapidly, you can collect lots of data about what tigers are eating, what leopards are eating. And this is important because the Chinese want to put these species back in areas that they're hoping to encourage tigers and leopards to recolonize. And also because they think that the tigers and the leopards are probably competing to some extent for prey. So they need to figure out how to manage these populations and encourage them back to where they used to live. So we've talked a little bit about how to collect data and the kind of complexity of the data that we're collecting. This is all very well, but we have to figure out how we're going to get those data to where they're needed. So how are we going to implement the strategies that we want to have on the ground? How can we protect small areas? How can we protect big areas? How can we do what Stuart's doing with saving species and try to buy areas of land that animals can move into and be protected, to repopulate land that's been degraded? And I think the big question here is engagement. We need to engage the wider community, not just conservation biologists. We need to engage everybody around the world who carries a smartphone or a drone or even has a camera and eyes and keen powers of observation in this process. So ConservationFIT is your project. You guys out there, many of whom have helped us in so many different ways, but also people who are listening in from outside today. This is your project. You make of it what you will. There are so many opportunities here to engage. And I think, the first, one of the most important is communicating conservation. Now, we do that partly at events like this. We have academic publications. We write in popular science. But communicating is, we need help with communicating to be able to reach out and explain to people that this project is happening. There are huge opportunities in data for good, in collecting data that can be pushed out there to tell people what's going on, to say, look, you know, this is data science, it's doing good in the world. I've talked quite a lot about deep learning R&D, but there's a lot of other uses that this data can be put to in research and development. We've done a lot of work with JUMP in mapping and in data visualization. And there are many other ways in which people with these skills can engage and have opportunities with the data that we've got. This picture was taken at Carolina Tiger Rescue this weekend. This is our most faithful team of SAS volunteers who go out on a regular basis to help us collect tiger footprints for our reference database. These guys go out in all weathers. They're fantastic. They've got the SAS flag flying there in case it rained last weekend. But there are many other opportunities to volunteer. We have a bird strike project on campus to try and stop birds colliding with all the glass that we have here. We've had other projects that citizen science can engage in, and there are many others that we could develop, not only here on campus, but around the world. STEM is a very big opportunity to engage with the project. We run a STEM project with SAS using animal footprints to take around to schools to show what can be done with science, technology, engineering, and math. There are many opportunities still awaiting for people at SAS and JUMP, perhaps who have children who would like to volunteer or get engaged with the project. And I think last of all, but certainly not least, we have a very wide range of global partners. Now, I think we operate in 16 countries at the moment, and that's going up all the time. I think we added Slovakia to that list yesterday, so it's 17. But we have global partners all over the world who really want to connect. They want to connect with the world. They would like to connect with SAS offices around the world, with other partners that we have around the world, to try to form a network of people who collect data and who analyze data. Huge opportunities there. So I think the bottom line is that this is your project. We would love you to engage. We want you to be part of it. We thank you for all the amazing work that you've done for us over the years. And many of you are here today. Many of you hopefully are listening in. Just to say a very big thank you for everything you've done to make this possible. And we look forward to taking it forward with you in the future.
Thank you, Zoe. I think we've got some time for some questions from our studio audience. Will you guys come back up, Skye and Stuart? And we've got someone who's going to run microphones because we've got the webcast going. Let's use a microphone when you ask a question, please, so everybody can hear. I wanted to start with one question. I believe that there are some teachers and some students, elementary, who are watching this today. What's your advice to them to get started with their conservation science? Any of you?
Well, I think that's a very good question, and I've had that question given to me several times this week as well. There are very many ways in which we have engaged with children in the past in schools, from simply researching how we use footprints and where they can be used, to actually going and picking up footprints, to doing bio-blitzes, where schools can go into an area and try and literally on one day identify all the species they can with experts. I would love to develop something involving children in data on that sort of level, as SAS does with STEM, or perhaps through JUMP or both, that we can actually show children, collect footprints, bring them in, here's how you analyze them. I think the subject matter in itself is very appealing to children. They relate to it. They can relate to it very easily. So lots of opportunities.
If I can just add to that, I think along those lines, SAS has been using the STEM work, regarding wildtrack, and it's proved to be one of the most attractive projects, because as soon as you actually try to explain data and analytics using animals, it's a fantastic combination.
A generation ago, maybe 2 generations ago, we were worried about our children spending all the time in front of the TV. A generation ago, we were worried that they were playing video games. And now if you watch children, they're engaged with these all the time. And yet this is a remarkable biodiversity assessment tool. You can send your kids out into the bush and they can photograph stuff. And it's engaging. It's a community of people who can be thinking about natural history. And I think that's wonderful. I think this is a very seductive device for young people, and to get them engaged in these efforts. First of all through iNaturalist, and then in time with the more sort of sophisticated things that we're doing with footprints.
Awesome. I think so too. Lander, okay, great question from Russ Wolfinger.
Hey guys, great presentation. I wondered if you could give us some more details about your database currently. How much space does it consume? And I was also curious, when you upload a footprint, is that also part of iNaturalist, or is the footprint database separate?
Do you want to answer that?
That's a good question. Well, the first thing is we're working with endangered species, so we don't have, at the moment, vast amounts of data for any single endangered species, and they're all held at wildtracks. We don't have a huge server for that. In terms of the data that goes onto iNaturalist, that is held on an iNaturalist server, but it's free for us to download again. So when people start uploading very large numbers of images, you're quite right, Russ, we will need a bigger platform to hold those images.
I'd like to comment on the security issues. One of the questions that we faced in the very, very early days of iNaturalist, when it was based in my lab, it's now based at the California Academy of Sciences, was how to protect confidential information. And there's basically a series of protocols. You can put your observation up and say, yes, I'm happy for people to know I saw this species exactly here. You can make those data fuzzed in some way. Typically, you put a location that's somewhere within 25 kilometers of that. And you can let iNaturalist only have access to those data. Or you can, in fact, say, no, nobody can have access except within this circle of uncertainty. Because absolutely, there are a lot of endangered species that we don't want to tell people exactly where we found them. So those kind of protocols were built into iNaturalist right from the beginning.
We've got a question right down here.
Thank you very much for such an interesting presentation, and I'm so proud and so pleased that this effort is going on globally. So thank you for the world. My questions are more of a layperson who's just fascinated by biology. So I would love to know, how can you tell species sexes from their footprints? And also, because of the material that the footprint is made in, because that varies so much, then how can you tell one animal from another?
Can I borrow your hand? We've done this many times before. Okay.
Go on, Stuart, you just want to hold a hand.
I just want to hold your wife's hand. So make sure I got the right hand. No, this hand is fine. Now, here are two hands. And I look at Zoe's hand and I deduce from that she is a female. And I deduce from mine that I'm a male. Look at the length of that finger versus that finger. Okay? Generally, in men, that finger is longer than that finger, and in women, they're more or less equal. It's a statistical thing. You guys do statistics. I don't need to go any further. The wonderful thing about that is you've probably all seen these images of cave paintings where people have taken pigment and blown a spray of pigment on the cave walls, and you get these handprints. And for a long, long time, anthropologists would say, well, this is where the men come in after the hunt and they bond in the caves and, they tell their stories. It turns out that most of the handprints are female. So there's a lot of subtle things. There's a really lot of obvious things once you begin to look. The other question I will hand to Zoe's husband.
Just to add to that, I'm sorry, I really apologize for running through that section very, very rapidly, but it was instructions from the boss. and you always listen to the boss. But basically what I should have explained is that we take lots and lots of measurements from each footprint based on certain concise and consistent points that are repeated. So we have, for example, if I was to take a hand, I know that point, I know that point, I know that point, and so on and so forth. So we establish what we call these landmark points on the footprint. We are trying to automate that system. It's extremely difficult. We've tried it time and time again, and it's come up with very, very varying results. But basically, it allows us to generate all these metrics. So for example, for the Amur tiger, we generate upwards of 125 different measurements, and they're in the form of distances, angles, and areas, all scripted into JUMP, so that once I put those points, those initial landmark points in, all the rest of the points are the metrics are generated automatically. And then it's a question of stats, and then you can actually apply fairly simple statistical methods like discriminant analysis, which actually distinguishes male footprints from female footprints.
Can I ask a follow-up question?
Absolutely.
So, do the tigers or the cheetahs or the other species that you're tracking, like human footprints are unique to each individual, is there something about each of those animals that distinguishes one individual from another unequivocally? Is it their stripes, or is it their footprints, or is it something else?
Well, as far as footprints are concerned, once again, the same analytical methods apply. We just run through all these different metrics, extract all these different metrics, and then we analyze them using, as I said, with this model that we've created, the customized model that we've created, which basically compares trails. So generally speaking, we don't just compare single footprints. We take a trail, and we've established what the sort of number that we need in each trail. And then we basically do a simple comparison, and it turns out that, yes, there are actually quite significant differences between individuals.
Great. Got some questions down here?
Hi, just wanted to extend that question just a little bit further. How do you tell, so every animal, hopefully, has 4 footprints, right? So if you have, say, like 13 trails of the tigers, how do you tell which right and left paw print are from which animal? I'm assuming they have relatively similar sizes and different individuals vary in that, but let's say 2 individuals of a similar size, how do you tell those 4 prints apart from each other?
So that's basically, that's something that comes with experience in walking in the bush and looking at footprints. Any good tracker will be able to tell you straight away, they know the difference between a left and a right, for example, simply by the way the toes are angled. So if you look at your own hand, you can tell the left goes, I can't tell left from right. This is left. The left goes down that way and right goes down that way. So, and the front are generally wider and shorter than the back. That's for carnivores. Now for elephants, the front feet are big round things and the back feet are more oval. It varies by species. There are some species that are really hard, right? We had a hard time with bongo, wasn't it? Hoofed animals are very difficult.
Yeah,
Antelopes are
very
difficult. So it's very much an individual species thing, but generally we're able to tell. And if we can't tell, then the algorithm can. So we'll actually collect known feet from a group of individuals, and then we'll shove that through the system. It'll say, okay, left hind, right hind.
And how do you tell that this left foot is from animal A and this right foot is from animal B? So if you have 13 different tracks, how can you?
Right.
Oh, I see. Now these, what happened was that these were picked up as trails. So an animal is walking along a trail. So for sure that's one animal walking along, creating that trail. And then you pick up another trail. If the animals are crisscrossing or they're overstepping and stuff like that, then of course the problem becomes extremely difficult. You can still do it. And we did this with white rhinos in Namibia when we set up the database. The whole database was set up using white rhinos. And white rhinos are much more social than black rhinos. They hang around in herds. So you can get up to 6, 8, sometimes even 10 individuals in a herd. So we had these herds moving together, but you could still follow certain individuals. So you could pick out certain individuals when they sort of veered off. So it is possible to do it, but it becomes increasingly difficult.
And once you determine all 4 feet from an animal, do you keep that as those 4 tracks for that animal? So you could say, oh, this print is from that animal, right? I'm sorry, I'm not perfectly clear. Do you keep each of those as a record, like each animal has its own record?
When we're developing the reference database for a species, which we do in the beginning for every species, we collect prints from known animals. We usually ask the collector to get all 4 prints, left and right and front and back. And we have those as our reference set. But we then generally choose 1 print that we're going to be working with for the footprint identification technique. And the reason for that is that we don't want people in the field to have to be recording every single footprint in the trail that they see. So with carnivores, we generally settle on the left hind print because carnivores generally do something called registering. Their left hind and right hind feet step on top of the prints made by the front feet. With bears, we often take the front feet because they tend to bring their back feet round and bring them over the front, and so the front feet are clearer than the back. So it varies a lot. Sometimes we go for a foot that shows more complexity. So in the giant panda, the front feet are much more complex than they are in the back. So we'll choose those. So it's very much a customized model for each species, simply because of the variety that the species exhibit.
Panda is really unique because it's got 6 digits. It's got a special digit, which is an extension of a sesamoid, what's called a sesamoid bone in the hand. And so now it gives us better complexity. And the more complexity there is, I mean, if I was to ask you, of all the animals that I had shown slides for with footprints, which was the most difficult animal to deal with? Would you like to hazard a guess? People? No, people would be easier. No, I'll tell you, it's the elephant. Because it's just a blob in the sand. And we work with dormice. I mean, we've actually set up a database for dormice to distinguish dormice from other mice. You know, so it's a species-based discrimination. And that's actually quite easy because they leave beautiful footprints, and these are ink prints. Okay? But when it comes to an elephant, you'd think that something that's going to measure 30 to 40 centimeters in length would provide you with the most fantastic amount of information. No.
Great. Let's do one more question, and then we'll wrap up the webcast, and I think we can ask our scientists to stick around a little bit longer if we've got any after that. But let's do one more for the webcast.
So my question is about the use of drones. It seems like that's an emerging tool for you guys to use in the collection and be able to collect even more data. But my question is, how do you determine the scale of a footprint if you're not there with a ruler to be able to sort of determine that on the spot?
That is a very good question. So basically there's no way you can, have someone running around on the ground with a ruler underneath the drone waiting for it to photograph the footprint. There are ways that you can estimate scale on the ground if you know the focal length of the camera on the drone and the altitude of the drone. But at the moment, with the sort of drones that we're using, these are not very accurate estimates. So what we're going to be doing at the moment is using the drones to identify trails and species, and then probably what we'll do is go to the ground and pick up the footprints manually. But it's possible that deep learning will find a way to circumvent this problem if we can identify the images without scale.
Thank you. I think it's time for us to wrap up. So I want to thank Doctors Jewell, Pim, and Alibi. Thank you so much. And then I will ask our audience, please go online and check out the new website. It launched officially yesterday. It's ConservationFIT.org. Start digging around and start adding your own data.
Brilliant.
All right?
Thank you.
Thank you.